Hi, welcome back to the studio. We are joined here on the sofa today by Nathalie Kingston and Kathy Stirrups, who've just given a wonderful presentation about NIHR BioResource, and also Yasuhiro Murakawa, who's just given us an amazing presentation about all the work he's doing in transcriptomics. So I'm firstly going to ask Natalie, we heard about the incredible work that BioResource is doing, but can you paint a picture for us about BioResource as a program? What's it all about? What's the impact you're trying to make? And how did it come about in the first place, if you can share that with us?
Well, the overall game is very much to benefit patients. So this is what we do. We have tried to specialize almost in cohorts of diseases. So the whole point is really to try to have a sizable cohort of patients with specific conditions so that you can have almost like more personalized or stratified medicine, and then trying to get better treatment, faster diagnosis. And we have already quite a few impact like this in the NHS, so in the UK, basically. But yeah, it all started very simply back in, I think, 2005, 2006 in Cambridge, UK again. Where there were some investigators, they wanted to look at a potential genetic variant in type 1 diabetes, and they started to bleed each other in the lab, and they realized that that was not really the way to do it. So started to actually wanted to engage members of the public, volunteers, and it all started from there and developed, and now probably around almost 350,000 actually participants. So long story, but yeah, a lot of potential, really.
Well, it was fantastic, and it was fantastic, Kathy, to hear about the scientific potential for this sample set. I'm wondering what it means for you as a scientist to see all this sort of broader genetic variation being brought into a sort of larger sample set for the benefit of the broader community. What do you think the impact of that will be?
So the way my career's progressed is that I facilitate other people's science, really, and that's what I can do through the BioResource. I keep building bigger and bigger data sets with newer and newer technologies. I know from our previous project how many publications we came out of that, big ones, small ones, methodology ones, patient studies, new genes being discovered. That's exactly what I want out of this new data set we're building with 22,000 genomes, or up to that many.
Amazing.
You know, there's plans we have it within our themes, but there's just so much that could be discovered using this data, and that's where I really want the whole international community coming in and being able to use it and discover lots of exciting things for the good of patients everywhere.
Science begets science begets science.
Yes.
Fantastic. Well, Yasuhiro, you also presented not only a sort of direct cDNA data set, but the most amazing direct RNA data set. And you said on stage just now that you're hoping for that data to be released to the broad community very quickly. So I think we're all interested in what the impact looks like for a Japanese data set to be released to an international community. What would you like to achieve by a broad data set release?
Yeah, thank you for the very great point. We hope that this atlas of gene models, I hope that can serve as a reference to understand somatic mutations and also genetic variations. But we also have data sets not only from Japanese samples, but also from international samples. So yeah, I think it's very important to look at diverse samples because, to understand the complexity and differences of the genetic basis.
What has the engagement looked like at RIKEN? How have you managed to build this broad sample set in a similar way to the BioResource one?
We also use purchase samples as well, and also cell lines. They also contain there are many samples derived from international efforts. So we also we work together with BioResource centers, trying to really make a very strong international samples.
Fantastic. Could you tell us a little bit about the international data sets? What's the gap that you're filling with transcriptomics, and in particular direct RNA? You spoke about the amazing work you're doing in sort of base modification discovery, and particularly in direct RNA. Is that work a big gap in the community at the moment?
I think the RNA modification is really something that we are starting to see. And we still, at this moment, don't know what the biological impact or clinical impact of them are. But I think with the clinical integration, the clinical information, and all this data needs to be integrated, like a multiomics-wise. And I think that would hope to help understand a lot more.
Fantastic. So we were talking just before the sessions about international collaboration in particular. What does it look like? Where do you travel? Who do you speak to about these international collaborations? I know, Kathy, you do a lot of that work.
Yeah, less so on the travel, but we have collaborators all over the world. The work I do on rare diseases, the important is about bringing in to people from all over the world, because these diseases are so rare. To get the sample sizes up to make the gene discoveries, you need to have those collaborations. So yeah, we're plenty in Europe, but also across America, even in South America, Australia. We work with lots of different groups around the world. And then the other thing we touched on is the importance of working with so many different ethnicities as well. It's in our talk how one of our latest cohorts is about looking at underserved populations and how we're really focusing on the black community, at least initially, and hopefully that will expand to other communities. And understanding that genetic background in the context of diseases is really important as well.
Well, we had Yor Bediako on this sofa about a couple of hours ago, talking about the fact that African representation in large-scale genomic databases is about 3%, which it's great that you're looking to address that disparity. What are the impacts on society for having a lack of representation in these data sets? What does it mean for you?
Well, for us, I think, in the UK, there are about 18% of the population who is not classified as white. So if actually you do not address some health needs, for those population, we are actually failing a lot of individuals. So Cassie mentioned the Improving Black Health Outcome, which is really dedicated for engaging with individuals from black communities. We've started a focus with looking at sickle cell anemia. It's a program with Genomic England as well we're working with. But we have about 2,000 now participants just in the last year. And that engagement needs to continue and expand. But having RIKEN and others who are also doing work, then we can actually fill those gaps, because we need to find the solution for the condition for everybody and not just for the majority.
So the more discovery you can do for the more people, the more methods we can develop, and the more diagnostics can be rolled out.
Absolutely.
Fantastic. Well, I want to move on to a slightly different topic, which is everyone at Oxford Nanopore who presents on the London Calling stage gets to pick their own walk-on music. It's a feature of the conference now. I know, Kathy, you had some really specific thoughts about your walk-on music, and I want to hear all about them.
Yeah, we probably overthought it a little bit. I had an initial idea, and then I looked a little bit more at the lyrics of the song, and I thought maybe not without explaining it. So then I opened it up to our team, and we had a fun afternoon trying to suggest songs. It turned out they were better at suggesting inappropriate ones. So, for instance, we've got the workflow demo in the afternoon, and you'll learn there how much we've struggled with the heat output of these Prometheans in the lab, and when you have a number of them running 24/7, it gets really hot in there. So we had Feeling Hot as a suggestion, which we didn't go with. In the end, Natalie was one who chose the song, and we went with one without lyrics. But for us, it picks up on the technological and innovative aspects of this technology. So it was a Jean-Michel Jarre electronic. Yeah.
Excellent. Great choice. Well, music and science go together so well. Well, before we go, I've got to say, our little birdie told me, Natalie, you are a racing driver on the side as well as being a scientist. So I'm going to ask you, which country or what location are you packing up a nanopore sequencer into the back of your car? Where are you taking it, and what are you going to do with it when you get there?
Yeah, I don't think I'm calling myself a race driver. I think your international source is not correct there. But I'm quite a petrol head. Yeah, that's fair enough. So actually in less than 3 weeks, I'm off to Le Mans, 24 hours endurance race, into a British sports car. So 8 to 10 hours journey there. It's going to get hot, so I will leave the Promethean in the lab because without aircon in my car, it's not that comfortable in middle of June. But yes, that's what I'm doing. And then I'm going to leave Kathy and the team keeping up the good work, and then I look forward to hearing more results when I come back. But yeah, this is the next trip.
Maybe you can put your Promethean in the car next time you go to a cold climate, and it will help you
I'll
along.
think about this, yeah.
Fantastic. Well, thank you so much, all three of you, for speaking on the stage and for joining us in the studio. It's been such a pleasure to have you and for you to share your work with the online community as well as the guys in the room. So thank you so much.
Thank you, Jane.
Thank you.